Hello. And so today, we're going to take a look at your protein and amino acid metabolism disorders. And so not all, but then just the ones that are actually common. Okay. And so let's take a look at your what you call this protein metabolism first. Okay. So with this, and so you have your protein turnover. And so your protein turnover accordingly here is the continuous degradation and resynthesis of your proteins. And so this one, there should be a what you call this for this, there is a degradation and the resynthesis. And so that degradation and the resynthesis should be equal. And so that is for your protein turnover. And so accordingly, this protein turnover is important. Ayan, so for the following. Ayan, so one, that is your metabolic flexibility is accordingly afforded by what you call this, relatively quick changes in the concentrations of your key regulatory enzymes. Other than that, you have your peptide hormones and the receptors for these enzymes and hormones. Again, and so you have here the metabolic flexibility of what they call this of a cell, and so is afforded by a relatively quick changes in the concentrations of your key regulatory proteins like your enzymes, your peptides, your peptide hormones, and your receptor molecules for this molecules for this regulatory enzymes and your hormones. Other than that, you have here your numerous physiological processes are accordingly dependent on timely degradative reactions and synthesis. Okay, and so you have here your numerous of physiological processes are dependent on timely degradative reactions as they are also on your synthesis. Okay, and then ayan, so you have here your proteins. Ayan, so your protein turnover. Ayan, so provide amino acid that is required for the synthesis of proteins. And so when nutrients are low, and this one also protects the cells from accumulation of abnormal proteins. Again, ayan, so it provides your amino acids that are required for synthesis of proteins. Ayan, so when your proteins are low, and then it also protects your cells from the accumulation of abnormal proteins. Okay, and so that is for your protein turnover. And so let's take a look at your protein degradation. You have actually listed here, actually you have two processes where your protein is degraded. One is by the action of your ubiquitin, or you have your ubiquitin-proteasomal system and your autophagy. Okay, and so let's take a look first at your ubiquitin-proteasomal system. And so accordingly, this is initiated by your by ubiquitination, and so that is your what you call this uh your ubiquitin. And so you have here your in your ubiquitination, ubiquitin is attached to a substrate protein, and so your ubiquitin here is a 76 residue protein that is 8.5 kilodaltons, ayan, in molecular weight. And so actually, it attaches to your substrate proteins. And so uh, the substrate proteins for your ubiquitin proteasomal system are relatively those that are short-lived proteins. Ayan, so they are your short-lived proteins. Again, ayan, so for the substrate protein, they are your worn out or damaged proteins. But then for your ubiquitin proteasomal system, ayan, so the substrate is your worn out or damaged proteins that are short-lived. Okay, and so they are short-lived. And so the substrate for your ubiquitin proteasomal system is your are actually proteins that are short-lived. Okay, and so accordingly, you have in these proteins, you have this marks or signs for proteolytic degradation. And so you call them as your degrons. And so these degrons are actually true in case of your ubiquitin proteasomal system. Okay, and so let's take a look at these digrons. You have actually two digrons that are listed here. you have here your degrons. And so you have here, you have your degrons. You have again your peptide motifs and your N-terminal residues. Accordingly, and so your peptide degrons or your peptide motifs, and so you have here your proteins accordingly with certain homologous sequences are rapidly degraded. Okay, and so examples that are given for this one is your proteins that have extended sequences containing your proline, your glutamate, serine, and threonine. They have accordingly half-lives of 2 hours. And so please look for your additional reading. And so please read on your half-lives of your proteins. Okay, and so you have there. Okay, and so again, for your peptide motifs, you have here uh, your proteins with certain homologous sequences are rapidly degraded. And then you have your N-terminal residues. And so for your N-terminal residues, and so you have accordingly your very short-lived proteins. Accordingly, your very short-lived proteins have basic or hydrophobic N-terminal residue. And so while comparing for your more stable proteins that have your that characteristically have your sulfur containing, your hydroxyl containing, or your non, or your what you call this hydrophobic amino acid at the N terminus. Okay, and so for your N terminal residues, and so your short-lived proteins have a basic or hydrophobic N terminal residue. And so comparing to your more stable one, those that contain your sulfur, your hydroxyl groups present in them accordingly. And so that is for your degrons. For your ubiquitin-proteasomal system, ayan, so it starts with your ubiquitination. Ayan, so your ubiquitination again, ayan, so that is the attachment of a small, highly conserved ubiquitin protein. Ayan, so that is the attachment, of course, is to your worn out or damaged proteins that are short-lived. Okay, and so that is for that. Your ubiquitination, and so accordingly, involves several stages. And so you have a several stages, and it involves three enzyme classes. You have your E1, and so your E1 here, that is for your ubiquitin activating enzyme. Of course, that is for the activation, no? And then for your E2, that is your ubiquitin conjugating enzyme. And so that is for the conjugation. And then your E3, that is your ubiquitin ligase. And so that is for the important enzyme classes involved in your ubiquitination process. Okay, and so after the ubiquitination process, and so here comes your what you call this, your proteins are transferred to proteolytic molecular machine, and so that is your proteasome. Okay, and so in your proteasome, and so this one actually is your proteasome is a protein again, and so that is a 200 sequence, and so let's take a look at your proteasome. And so right after ubiquitination, and so the protein is transferred into your proteasome. And so relatively, this one is larger comparing it to your ubiquitin protein. And so your proteasome, and so what does it do is, it cleaves your peptide to a should be, and so please change that one, and so it cleaves the peptide, and so it divides the peptide. And so suppose you have a long peptide, you have a long protein, you have the peptide chain, and so what will your proteasome do is it will chop, no, and so it will yes, it will divide, it will chop, no, and so you have here, it will chop, it will cleave, and so it will divide it will break down, or will cleave your peptide into at least a chain of 7 to 8 amino acid residue. Okay, and so 7 to 8 amino acid residue. Okay, and so once the protein is degraded, and so what you call this, you have here, once the protein is degraded, and so the short chain products are degraded by your, they are actually released, and the short chain are degraded by your cytoplasmic proteases. Okay, and so once the protein is degraded, and so and at least a 7 to 8 or probably 6 to 10 residue peptide products are released from your proteasome, and so they are what they call this release for hydrolysis of your cytoplasmic proteases to produce a free amino acid. And so, of course, after from your proteasome, and so they are your 6 to 10 or your 7 to 8 amino acids are are released into the cytoplasm and is acted upon by your cytoplasmic proteases to produce free amino acids. Okay, and so and then of course those free amino acids will go into your amino acid pool. Or probably they will be resynthesized again. Okay, and so that is for your ubiquitination, your ubiquitin proteasomal degradation. Okay. Than your ubiquitin-proteasomal system, you have your autophagy-lysosomal system. Okay, and so with this, of course, your autophagy, that is your self-eating. And so accordingly, this is a cellular degradation pathway in which your cell components, and so most for this, and so you have your long-lived proteins. And so unlike for your ubiquitin, proteasomal system, and so the substrate is the short-lived. And so with this, you have your long-lived proteins and your organelles, and so you have there. And so with this, of course, you have here the use of your hydrolytic enzymes present inside your lysosomes. Okay, and so you have here your hydrolytic enzymes present in your lysosomes. They are the ones responsible in degradation. Okay, and so again, they degrade your worn out or damaged cell components. Okay, and so with this, and so your autophagy, and so provides a recycling mechanism. And so again, they degrade your worn out or damaged cell components. And so with that, and so your autophagy provides a recycling mechanism. For that, and so to maintain your vital functions within the nutrient level. Yeah, so that is for your recycling mechanism. And then you have here your regulation and development as well as destruction of a microorganism. And so your autophagy accordingly is involved in regulation and development, probably for your cellular modeling, and of course the destruction of your invading microorganisms. Okay, and so that is for your autophagy-lysosomal system. Ayan, so you have your recycling mechanism, and then your regulation and development and destruction of microorganisms in your autophagy. Of course, this one is carried out by your lysosomes, and then of course the lysosomal hydrolytic enzymes present inside this vesicle. Okay. Okay, and so accordingly, you have your three forms of autophagy. And so you have your chaperone mediated, your microautophagy, and your macroautophagy. And so accordingly, your macroautophagy here, and so that is referred to as your autophagy, no? And so you have there, and so this one is referred accordingly as your autophagy. But then for us, and so let's call it as your macroautophagy. And so you have here accordingly for your macroautophagy, and so that is the use of your lysosomal pathway for a large scale degradation of some cytoplasmic components. And so that is for large scale or bulk uh, degradation of cytoplasmic components. Accordingly, and so it is the major catabolic mechanism that is used by eukaryotic cells to maintain an optimal function and to respond in a changing environmental condition. And so that is for your microautophagy. Again, and so that is used as it is the major catabolic mechanism used by eukaryotic cells, by your probably for us the animal cells, and so to maintain optimal function and respond to changing environmental conditions. Okay, and so and then you have your micro autophagy, and so for this micro, and so this is a small scale naman, and so you have it as small amounts of cytoplasm are directly engulfed by lysosomes. And so comparing it, and so your macroautophagy, and so that is your large scale or your bulk degradation. And then you have your chaperone mediated, and so your chaperone mediated, that is, you have your uh, is what you call this, your chaperone mediated as a receptor mediated. And so this one is a receptor mediated process in which you have your specific proteins that are bound to a chaperone, and so that are bound to a chaperone complex are unfolded and then translocated in the lysosome. Ayan, so in the lysosome, ayan, so they are unfolded and translocated into the lysosome. In the lysosome, that is the site where they are degraded by lysosomal hydrolytic enzymes and proteases. Okay, again, so you have your chaperone mediated, your microautophagy, and then you have your macroautophagy for that. Okay, look at your autophagy again. And so your autophagy, this one is induced by your stressors. And so you have actually a large number of stressors. And so that includes, you have here your hypoxia. Ouch. Sorry. You have your hypoxia, oxidative stress, nutrient deprivation, and then you have your high temperature and together with your viral infections. And so among others, of course, with that. Okay, and so they, what you call this, they induce, and so this uh these processes and so induce your autophagy. Okay, and so your autophagy, of course, begins with the formation of your autophagosome. And so in case of your ubiquitin proteasomal system, and so that is the attachment of the protein ubiquitin. But then for this, and so you have your autophagosome, and so that begins with the formation of your autophagosome. Okay, and so this autophagosome, ayan, so then fuses with an endosome to form an amphisome. And so it will fuse with an endosome to form an amphisome. And so you have your endosomal membrane bound ATPase. ATPase accordingly begins to pump protons into the amphisome lumen. Ayan, so with that, eventually, ayan, so this amphisome fuses with your lysosome to form your autophago-lysosome. Okay, and so to form your autophago-lysosome. And then, of course, in you have there in once there is already a formed autophago-lysosome, then what you call this, you have there the action already of your lysosomal enzymes, and so they proceed to digest the cytoplasmic the cytoplasmic package, and so they yes, and so this what they call this, the proteins inside the autophagolysosome, and so and they are digested, and then after being digested, and so they are released. And so the degradation products and so are exported to the cytoplasm. And so they are exported into the cytoplasm where they are used in biosynthesis or they are degraded to generate energy. Okay, and so in case of, let's say, you have your amino acid, you have your sugar, and so the amino acid, and so is used for synthesis, and the sugar that is degraded for energy. But then, and so accordingly, there are in your autophagolysosome, there are residues, residual bodies, and so those that resist digestion. Okay, and so what happens then with them, and so they remain in the cytoplasm, and so they remain in the cytoplasm, giving you your cytoplasmic inclusions like your lipofuscin granules and your pigments also. Okay, and so those items for your autophagy. Okay. And so let's take a look at your what you call this protein. And so generally for your protein deficiencies, just some. And so you have your muscular dystrophies. Accordingly, these are actually a group of inherited diseases, no? And so this one is characterized by progressive muscle degeneration and weakness. Okay, and so this one is characterized. This is this your muscular dystrophies is actually a group of diseases, and so you have many for that. And so you have that is characterized by progressive muscle degeneration. That is probably the the hallmark of this one. Okay, and so you have here accordingly, and so your dystrophin, and so your dystrophin, and so the dystrophin protein, and so that is actually the affected one, and so that your dystrophin accordingly, and so anchors, and so normally the dystrophin anchors the intracellular cytoskeleton to the extracellular matrix, and so with that, and so they anchor, and so your intracellular cytoskeleton to the extracellular matrix helps to transmit force during muscle relaxation. And so in case, ayan, said that there is no such thing, and so then there is what they call this, a what they call this, a problem in muscle contraction and relaxation. Ayan, so with this, ayan, so your proper metabolism again relies on the integrity of your cellular structure, and your dystrophin plays a crucial role in maintaining the integrity of your cellular structure. And so that is for your muscular dystrophy. And so you have actually your Duchenne type, your progressive type, and the rest. No? And so that will be discussed to you in your other subjects. Then you have your amyloidosis. And so your amyloidosis is a group of disorders characterized by extracellular deposition of abnormal protein. Abnormal protein fibrils known as your amyloids, no? And so these are your amyloids. And so they are deposited in your various tissues and organs, no? And so you have there. And so these amyloid deposits accordingly, and so will you can has the ability to interfere with the normal tissue structure and function. And so with that, it will lead to a range of clinical manifestations. No, and so it involves your production, and so it affects the production, misfolding, and accumulation of specific proteins with this. Okay, and so your amyloidosis again is associated with with misfolding, and so for that, and so you have there a common sites of deposition for your amyloidosis. You have your kidneys, your heart, your spleen, your liver. Ayan, so you have there. And again, your amyloid deposits interfere with the normal architecture and function of the affected tissues. Okay, and then of course, it can cause cellular toxicity also and inflammation. And so by the presence of your amyloid fibrils. Okay, and so that is for your amyloidosis. Okay, and then you have your kwashiorkor, no? And so your kwashiorkor with this naman, and so this is actually a severe form of malnutrition, no? And so this is characterized by a protein deficiency. And so this one typically occurs in children, no? And so your kwashiorkor, and so this is primarily caused by a diet deficient in proteins. And so that is, you of course, they are essential for the synthesis and synthesis of critical proteins in the body. And so including those involved for the fluid balance and immune function. Okay. And so you have your for your protein deficiency, and so of course you have your causes inadequate amino acids. And these amino acids, they are precursors for the synthesis of other materials, no? Other materials needed for normal functioning. And so you have here your protein deficiency leads to an inadequate intake of essential amino acids. And then of course these amino acids again are crucial for the synthesis of proteins involved in uh, physiological processes. Ayan, so, and then of course, once there is a the protein is deficient, ayan, so there is a large scale protein deficiency, then it affects your body's ability to maintain normal cellular function. Okay, and so it affects growth, ayan, so immunity with that. And so the skin, ayan, so it affects multi-organ actually for this kwashiorkor. Okay, and so this one, and so is typically seen in areas of severe poverty also. Okay, and so that is for your kwashiorkor. And so you have your phenylketonuria. And so with this, and so for your phenylketonuria, and so for your phenylketonuria, ayan, so this one is a deficient, I mean, a what you call this, a amino acid metabolism disorder naman. And so, and the rest that we'll be talking, no? And so this one is a condition that is a what you call this caused by a deficiency of phenylalanine hydroxylase. And so this one is a deficiency of your phenylalanine hydroxylase. And so with this, and so actually, and so your PKO, your phenylketonuria, I think, is part of your expanded NBS. No, and so with this, and so if this condition is not identified and treated immediately after birth, then it can cause mental retardation and irreversible brain damage. Okay, and so this damage and so results mostly from and so your what you call this your brain damage, and so this damage results from your mostly from the accumulation of phenylalanine. And so your phenylalanine, because your phenylalanine is not metabolized, and so andyan lang siya. Okay, and so your high phenylalanine blood levels results actually in the saturation of the transport mechanism for your large neutral amino acids across your blood-brain barrier. That is why it causes this causes a brain damage. And so there is a decreased levels of protein and neurotransmitter synthesis. And so due to what you call this, there is no transport, there is what you call this, saturation of the transport mechanism for large neutral amino acids. Okay, and so you have there your phenylalanine is transaminated, and so in the presence actually, and so in the presence of your uh in the absence, and so in the absence of your phenylalanine hydroxylase, and so your phenylalanine is transaminated to produce your phenylpyruvate. Okay, and so your phenylpyruvate, and so your phenylpyruvate is converted to phenylacetate and phenylactate. Okay, and so your phenylpyruvate is converted into your phenyllactate and phenylacetate. And this phenylacetate and so gives the urine the musty odor. And so this one actually, this phenylacetate is excreted. A large amount of this phenylacetate is excreted in the urine. And this phenylacetate and so gives the urine its characteristic musty odor. And so that is your musty odor that is characteristic, that is for your phenylketonuria. And so that is for your phenylketonuria. Okay, and so and your phenylketonuria for the treatment accordingly is uh, treat with a low phenylalanine diet. And so that is your for your phenylketonuria, and so treat with a low phenylalanine diet. Your alkaptonuria. Alkaptonuria, and so that is actually, and so your alkaptonuria actually is the first disease, and so to be linked to a genetic inheritance, and so that involves a single enzyme. Okay, and so this alkaptonuria is caused by a deficiency of your homogentisate oxidase. Okay, and so this one is an enzyme. Your homogentisate oxidase is an enzyme required for the catabolism of your phenylalanine and the what you call this probably the aromatic ring present in your phenylalanine and tyrosine. Okay. Ayan, so your homogentisate is actually excreted in the urine in large quantities. Ayan, so upon oxidation of a urine that has a homogentisate, ayan, so it turns black. Ayan, so it turns black accordingly. Ayan, so you have there. Okay, and so it turns black. And so you have for that one. And so with this, and so your and so accordingly, and so the black urine appears to be benign. And so, but then, and so alkaptonuria accordingly is not innocuous. And so because your what you call this alkaptonuric patients develop arthritis later in life, no? And so your what you call this, there is a development of arthritis later in life of the patient. And then of course, in addition to that, there is an accumulation of pigment. And so what you call this, accumulation of pigment that will cause uneven darkness. I mean, uneven. I mean, the accumulation of the pigment causes unevenly dark. Yes, uneven distribution of the pigment, causing unevenly darkness of this it unevenly darkens the skin. Sorry. Syrup urine disease. And so this one is also called as your branch chain ketoaciduria, no? And so this one is also called as your branch chain ketoaciduria. And so your alpha keto acids derived from your leucine, valine, and your isoleucine accumulate in large quantities in the blood, no? And so accordingly, and so it imparts a maple syrup odor in urine. And so it imparts a maple syrup urine odor, probably a sweet, no? And so sweet odor. Okay, and so accordingly, and so all three alpha keto acids accumulate because of a deficiency in your this one, your branch chain alpha keto acid dehydrogenase complex. And so you have the deficiency of your branch chain alpha keto acid dehydrogenase complex. Okay, and so accordingly, and so if what you call this left untreated, because what sorry, and so your this alpha keto acids accordingly are what you call this converted into acyl CoA derivatives. But then in the absence of that branch chain alpha keto acid dehydrogenase complex, and so they are accumulated, no? And so accordingly, if this uh MSUD is not treated, and so it can cause uh mental retardation, no? Vomiting, convulsions, and severe brain damage. Okay, and so your patients that are untreated and so often die before 1 year of age. And so the treatment for this one is same with your phenylketonuria, and so as what we call this a dietary control pa rin. Okay? And so that is for your maple syrup urine disease. And then you have your methylmalonic acidemia. Your methylmalonic acidemia, ayan, so that is caused by the deficiency in methylmalonyl-CoA mutase. No, and so you have here methylmalonyl-CoA mutase is absent, deficient in methylmalonic acidemia. And so this actually is involved in the conversion of your propionyl CoA into succinyl CoA. Ayan, so you have there. And so in this, and so there is in your methylmalonic acidemia, there is an accumulation of your methylmalonate in the blood. No, and so there is an accumulation of methylmalonate in the blood. And so its signs, its symptoms are similar to those of your maple syrup urine disease. Okay, and so the symptoms are similar with your maple syrup urine disease, and your methylmalonate may also accumulate because of a deficiency of your adenosylcobalamin. And so your methylmalonate can also accumulate because of a deficiency of your adenosylcobalamin. Okay, and so you have there. And so your adenosylcobalamin. And so with this, and so accordingly, a dose of some individuals, and so respond to injections of large doses of vitamin B12 daily. And so they respond, and so for what you call this uh to avoid accumulation, then probably, and so yeah, the introduction, and so the injection of vitamin B12, and so probably large doses daily. Okay, and so that is for your methylmalonic acidemia. Then you have for your albinism. And so you have your albinism. And so that is your albinism actually is a genetic defect again. And so that is albinism, a genetic defect with serious consequences. And so this one is caused by a deficiency in your tyrosinase. And so your deficiency in your tyrosinase causes albinism. And so with this, you have your melanin, and so consequently, accordingly, your melanin, that is a black pigment that is present in the skin, the hair, and the eyes, are formed from tyrosine in actually several types of cells. And so in what we call this in albinism, ayan, so melanin is not produced. And so you have here tyrosinase accordingly. And so with that, in the presence of tyrosinase, you have here tyrosinase converts your tyrosine to L-DOPA and then L-DOPA to dopaquinone. And so, and then of course this one, and so will condense to form melanin. But then, and so in the absence of tyrosinase, and so there will be no melanin that is formed. And so giving you what we call this a lack of pigment, and so giving the term albinos, no? Albinos to individuals affected with albinism. And so they are those individuals with albinism, they are extremely sensitive to sunlight. And so, and they have what they call this a susceptibility to skin cancer and sunburn. And accordingly, they have a poor eyesight. And so that is for your albinism. Okay, again, and so there is a deficiency in tyrosinase. Okay, and so that is for your protein metabolism and amino acid metabolism disorders.